The next company to present is AcouSort. Welcome, CEO Torsten Freltoft.
Thank you very much. And thank you very much for inviting me. I'm going to talk about how cell therapy can become mainstream. And I was very happy this morning to hear that this is something that is really important in today's discussion of cell therapies. But before actually going into that, I would like to acknowledge all the fantastic innovations that have been presented or will be presented here today. It's fantastic to hear about this many companies that are contributing to cell therapeutic developments. However, for all of these companies or the cell therapies that they're developing, it will take something to make them mainstream. And mainstream meaning that they can be provided not to thousands of people or hundreds of thousands, but hundreds of millions. And this will take something else. It will take developing picks and shovels for this type of technology. And what do we mean by picks and picks and shovels? Picks and shovel investments or picks and shovel infrastructure is a term that came out in the 1850s when the gold rush was hitting California. And so investors at that time could choose to invest in special teams that would go out and equip them to dig gold and try to find gold and become rich and get a return on their investment this way. But some companies chose to not do that. They chose to sell picks and shovels or all other types of gear that was necessary for all of the teams to become efficient. And those were often the investors that actually got a good return on investment because the risk there is a lot lower. Now, let's just talk about what picks and shovels actually means in today. This is a picture of sort of typical cell therapy labs. And as you can see, lots and lots of picks and shovels more advanced than the old ones are required to do the research and the development and also the treatments that are already in clinic. You need pipettes, centrifuges, microscopes, syringes, glassware, flow cytometers, sterile plastics, test tubes. The list is very, very long. And that's, of course, very good. But this is still very expensive and very labor-intensive. Let's take an example. We talked about CAR T cells. It has been mentioned several times this morning. And, as it has already been explained, you take cells out of a patient, you train them to recognize the cancer, you expand them to get lots of cells, and then you re-inject them into the patient. And as we've heard, there are lots of issues or lots of challenges. But what we haven't talked too much about is that to do this, you have to actually work a lot with these cells and process them in very different ways. You have to separate the immune cells from the rest of the blood. You have to enrich the cell population of interest. You have to provide access for viral vectors to actually implement the training. You have to wash away now the viral vectors again after. You have to expand them. You do that in incubators and where you have growth media, so you multiply the cells. Here, of course, after that, you have to wash away the growth medium. You have to up-concentrate the cells, and if they have been frozen to be transported, you have to eventually thaw them and wash away the freezing medium before you can actually re-inject those cells into the patient. Now, all these processes, they are done mostly manual, and also with a high risk of contamination during the course. And to mitigate these risks, it becomes a very expensive process to do all this. and that has to be overcome before mainstream can be achieved. This is where AcouSort comes into play. So let's talk a little bit about my company. AcouSort is developing components that can do these processes in an automated way. So what we will do is produce and supply consumable OEM separation modules that can be built in to third parties' closed systems or closed or automated systems that will handle all these processes. So here we have to the lower left an AcouWash OEM module, as we call it. It can wash cells, put them in a different medium, wash away contaminants, and it can up-concentrate cells, and it can also do some cell separations. We have an AcouTrap module where you can do, on smaller cell batches, many of the processes, and we have an AcouPlasma optical, as we call it, where you can get access to monitor the conditions of the cells or the background medium that they are in. How does it work? It works by combining microfluidics and ultrasound. And I have a small video here where it's just relatively simple. You have blood coming in from the left in the video, and then when you turn on the ultrasound, then you focus all the red blood cells to the center, and they come out in one exit, and the rest of the sample will come out in the on the side outlets. Now we turn off sound again, and you can see it's all over the place. This is just an example of one of those components and how the technology works. We also have two Benchtop systems that we use where we have implemented these OEM modules, and they are provided to our collaborators or to researchers that would like to have access to the type of sample processing in their development processes. So we work with big life science companies that will build the systems for these processes, and they can then have early access to the way we handle our samples through these systems. So our role in cell therapy, just to wrap that up, is enabling sample processing and integration and automation by eliminating this need for manual sample processing. And our technology fits really well into the steps of these processes. We can purify or isolate cells to ensure that only the correct cells are used in preparations. We can wash away contaminants from samples to ensure that the quality of the sample is maintained. And we can up-concentrate cells so that the cells can come and be also re-injected in the right concentrations, or in the right numbers. Right now, our initial focus is to ensure AcouSort's technology is further matured so that it can meet the sample volume requirement that is here in this cell therapy space. We have our original space, you could say, is focusing on diagnostic, and here we don't need to process quite as much sample to provide the sample processing required. But in cell therapies, these requirements are slightly higher. And we are really successfully pursuing this, you could call it, adaptation to the requirements in the cell therapy space at the moment. So on the business development front, I want to emphasize that we have ongoing collaborations or discussions with a handful of potential partners. It's amazing how fast these big life science companies now, based on the input we got this morning about new cell therapies being approved, that they pick up their interest in providing this mainstream ability in this space. And they share some of the visions that I list here. They would like to think in bedside treatment preparations. They want to decrease the time to treatment, the time to do the entire process. They focus a lot on robust cell processing, and what's maybe most importantly, they focus on significantly decreasing the costs of these type of processes or therapies. So, why would anyone invest in AcouSort? Well, to me at least there are 4 really good reasons. We represent a highly scalable business model with a recurrent revenue from consumables, our picks and shovels of the future cell therapy space. We have a validated technology coming out of Lund University. It has been developed within Lund University for more than 20 years. AcouSort has done the more industrial development for almost or just over 10 years now. And thirdly, there is this very significant interest from life science companies in both our 2 fast-growing markets, cell therapy and diagnostics. And at least at the end, we have the opportunity to enable the development of the next generation diagnostics and therapeutics, and in this way, providing more and better healthcare faster. And with this, I would like to thank you for listening, and I hope there are some questions. Thank you.
Thank you, Torsten. To all of our viewers that are not very familiar with the commonly used cell sorting methods, could you just elaborate on the advantages of AcouSort's technology compared to the commonly used methods?
Yes, absolutely. For quite a few years, you have been able to sort cells using something called FACS or MAX. And these technologies are really effective, but they all rely on labeling your cells of interest, and then for the FACS, it's fluorescently labeled. So you have to, so when you shine laser light on them, they can fluoresce, and you can then use this instrument to pick out those that are fluorescent and the ones that are not. And the other one is using more or less same type of approach, just with magnetic beads. So the magnetically labeled ones can be pulled away by a magnetic field. We sort cells, or can sort cells, if they have a different size or different density, just by the physics of it, and the physics between the sound, ultrasound we have, and the size or density of the particles.
So to summarize, so if you kind of limited the step where you need to use fluorescence and label the cells, you will most likely save a lot of time.
Yeah, I mean, especially when you can do bulk sorting just on the physical properties, that is the best way. And red blood cells and white blood cells at least can be sorted that way.
Perfect. So you mentioned your collaborations, and just recently you communicated extended collaborations in terms of cell therapies. So what can shareholders expect in from a short, mid, and long-term perspective from this collaboration?
Yes. You could say, I mean, these collaborations, they usually always come in different phases. I mean, initially we have some informal discussions about their interest and our capabilities. But then we enter usually some feasibility studies, meaning that now we have to show in the real lab that our technology can provide exactly the features and meet the conditions of this specific application. So that's basically where we are now. So further results along that line would be the short term. Then, if everything goes well, then we would start to work in more formal product development, where this would be sort of the second phase, and then of course the long term would be the commercial phase, where we would produce and provide these OEM consumables for our partners and then they would use them in their final systems that they sell to the end users.
I understand. And continuing on collaborations, because 2 weeks ago you also announced an extension of your AcouSort's Japanese collaboration. Yes. can you tell us more about that collaboration and the aim of it?
Yes, I can talk a little bit about it. This has nothing to do with cell therapy, so it's in the clinic, the diagnostic space. But we have worked with this Japanese company for more than 3 years now, and they have been developing an assay for a cancer, for cancer, so that they can diagnose a certain type of cancer using platelets. And they needed very clean samples from blood samples where they could extract the platelets without getting contaminations from any of the other cells. And they tried to do that by centrifugation approaches, but didn't work out. So with our technology, they found that here's the opportunity. And so we provided them with an AcouWash for this period of time. And now they finished the development of the assay and are now testing the assay on clinical samples to see how it pans out. And of course, we don't know what the result will be, and also we don't know anything about what will happen in the company after, because it depends on their internal priorities and so. But we've at least had a fantastic collaboration with them so far, and a very successful one seen from the technology point of view.
So just, I have a follow-up question, and I completely understand that you cannot provide us with detailed answer, but I'm quite curious, and I will give you like checkbox answers, and you can choose among those. So when do you believe the first product with AcouSort technology will be launched, and will it be like, do you believe it's 1 to 3 years, or 3 to 5, or over 5 years?
We've had a collaboration with a partner in the Boston area for some years already, and we hope and believe that this will actually be commercialized and hit the market already this year.
Wow, that's
fantastic.
And also with regards to future potential royalty agreements with global life science companies in the cell therapy space, what would be the strategy in terms of exclusivity and scope?
Yeah, that's a very good question. It's a natural reaction from our partners to say, well, now we work with you on this. Could we get some exclusivity, because it will, keep us in the lead when we start commercializing. And of course, I mean, if we are a picks and shovel company, we don't like exclusivity, because we want to sell our picks and shovels to all players. That's our plan A, at least. But of course, I mean, it is a matter of price. So if you come and say you need or want exclusivity in a very narrow area and for some time-limited period, and the price is good enough, I mean, I'm not ruling out that we would go into this discussion. But our main strategy is to keep away from exclusivity.
And just another follow-up question, because OEM products, and you mentioned that you aim to sell parts to other companies that do have the equipment already. Because I'm kind of thinking about the car industry, when every kind of, you have Volvo, they had their standards, and Audi, and they have their standards. So how do you see that? Is that an issue, or?
I think we will need to adapt the way our core technology is packaged and interfaced, but the real core, I mean, where the actual ultrasound meets the channels where they meet the sample, is going to be not very different. But of course, you're right that it may, if you have a big closed system handling many samples at the same time, or if you have a small bedside sample or bedside system, of course, the way it's sort of packaged is definitely going to be different. But it's not a matter of a big change or big difference.
To summarize the presentation, what would you say shareholders should remember from this presentation?
I absolutely think you should remember the picks and shovel idea, we provide the picks and shovels for the future of cell therapy. And in this way, we hope to provide more and better healthcare faster. Thank you.
Thank you.